Rahman Rahim. Today we'll discuss about reticular formation. Reticular formation. Reticular formation consists of groups of neurons and nerve fibers. Groups of neurons and nerve fibers in midbrain, pons, and medulla oblongata. So groups of small neurons and nerve fibers in midbrain, pons, and medulla oblongata. Reticular formation has ascending pathways and descending pathways. Ascending pathways and descending pathways. Reticular formation is connected to the cerebral cortex superiorly. So superiorly, reticular formation is connected to the cerebral cortex. Inferiorly, reticular formation is connected to the interneurons in the gray matter of the spinal cord. Inferiorly, reticular formation is connected to the interneurons in the gray matter of the spinal cord. In reticular formation, two areas are important. So two important areas in the reticular formation. First one is bulboreticular facilitatory or excitatory area. So bulboreticular facilitatory or excitatory area. And the other area is inhibitory area. Bulboreticular inhibitory area. Now first about the bulboreticular facilitatory or excitatory area located in the midbrain and pons. In the midbrain and pons, there is bulboreticular excitatory or facilitatory area. From this area, nerve fibers arise, and these pass to the spinal cord as reticulospinal tract, and nerve fibers also go to the brainstem nuclei as reticulobulbar tract. So reticulospinal tract and reticulobulbar tract, these carry impulses to the spinal cord and also to the motor nuclei in the brainstem. From this bulboreticular excitatory area, nerve fibers arise, which go to the cerebral cortex. So these nerve fibers arising from bulboreticular excitatory area and going to cerebral cortex, these nerve fibers constitute the reticular activating system. So these constitute the reticular activating system, the nerve fibers which arise from bulboreticular excitatory or facilitatory area, and these go to cerebral cortex. So these nerve fibers constitute the reticular activating system. Now most of these nerve fibers which arise from the bulboreticular excitatory area going to cerebral cortex, these first go to the thalamus. And then from thalamus, these go to the cerebral cortex. There are two types of nerve fibers which pass from the bulboreticular excitatory area to thalamus and then to cerebral cortex. Two types of fibers. First type are rapidly conducting nerve fibers. Rapidly conducting nerve fibers of large neurons in this area, And these nerve fibers release acetylcholine at the nerve endings. These release acetylcholine at the nerve endings, and these maintain excitability of the cerebral cortex for milliseconds. So rapidly conducting nerve fibers which carry impulses from this area to cerebral cortex, these release acetylcholine, these release acetylcholine at the nerve endings, and these maintain excitation of cerebral cortex for millisecond. The second type of nerve fibers are slow conducting nerve fibers. So second type nerve fibers are slow conducting nerve fibers. These go to thalamus. In the thalamus, these nerve fibers synapse in the intralaminar and reticular nuclei. So nerve fibers are slow conducting, and these synapse into the intralaminar and reticular nuclei in the thalamus. And from these nuclei, nerve fibers go to the cerebral cortex. So these nerve fibers are slow conducting, and these maintain excitation of cerebral cortex for seconds to a minute or so. So these nerve fibers are slow conducting, and these maintain excitation of the cerebral cortex for seconds to a minute or so. These nerve fibers maintain background excitability of the cerebral cortex. So these nerve fibers maintain background excitability of the cerebral cortex. How is excitation of bulbo-reticular facilitatory area maintained? How is excitability of bulbo-reticular excitatory area maintained? The first mechanism is sensory input of impulses. Sensory input of impulses, especially the pain impulses. Keep in mind that collaterals from all these sensory tracts go to reticular formation. Collaterals from all the sensory tracts, these go to the reticular formation. So sensory input of impulses maintain excitability of the reticular formation, or you say bulbo-reticular excitatory area. So input of sensory impulses, especially the pain impulses. You see, this is the entry of the fifth nerve, trigeminal nerve. Right? Entry of the trigeminal nerve into the bones. Now if we give cross-section in the brain, above the level of the entry of the fifth nerve, say here, in the animal, coma results. In the animal, coma results. Keep in mind the entry of the fifth nerve, trigeminal nerve, into the bones is the highest point at which sensory input enters the brain. So entry of the fifth nerve is the highest point at which sensory input goes to the brain. So if we give a transaction above the level of entry of the fifth nerve, coma results in the end. And if brain transection is below the level of the entry of the fifth nerve. There is no coma, no unconsciousness in the animal because the sensory input to fifth nerve, it goes into the brain. So if transection is below the level of the entry of fifth nerve, sensory input goes to the brain. So no coma results in the animal. So this shows the importance of the sensory input into the reticular formation of bulboreticular excitatory area to maintain excitation of this area. The second mechanism to maintain excitation in this area is feedback signals between this area and the cerebral cortex. So feedback signals between this area and cerebral cortex. Whenever brain becomes active because of thinking or motor process, from the brain, from the cerebral cortex, impulses go to this area. So this area become activated. So impulses go from this area to the cerebral cortex. So a vicious cycle of feedback impulses start between the cerebral cortex and this area. So vicious cycle starts between the bulboreticular excitatory area and cerebral cortex, which activate this area and also the cerebral cortex. You also should keep in mind that there are connections between cerebral cortex and specific areas in the thalamus. There are connections between cerebral cortex and specific areas in the thalamus. So this was about the bulboreticular excitatory area and the reticular activating system, which maintain excitation of the cerebral cortex, or which maintain wakefulness or alertness. The other area in the reticular formation is bulboreticular inhibitory area. Bulboreticular inhibitory area. This area is present in the medulla oblongata. When this area is stimulated, there is inhibition of bulboreticular facilitatory area. So when inhibitory area is stimulated, it results into inhibition of the bulboreticular facilitatory or excitatory area. Right? Inhibitory area is excited by a center above the level of inferior colliculus. Inhibitory area is excited by impulses from a center above the level of the inferior colliculus. So this was about the inhibitory area or bulboreticular inhibitory area. Now we come to the functions of the reticular formation. The first function is reticular formation through reticulospinal tract. Control activity of the alpha and gamma motor neurons. Muscle tone, especially in the extensors. muscle tone, especially in the extensors, and reflex activity. So reticular formation through reticulospinal tract control activity of the alpha and gamma motor neurons. Skeletal muscle tone, especially in the extensors, and also the reflex activity. The next function is that reticular formation contains vital centers. Reticular formation contains the vital center, vasomotor center, and the respiratory center. Vasomotor center and the respiratory center. Next function is reticular formation has a role in the control of visceral and somatic sensation. It has a role in the control of visceral and somatic sensation. I told you that collaterals from all the sensory tracts go to reticular formation. Especially pain impulses go to, slow pain impulses go to the reticular formation. So it has got a role in the control of the visceral and somatic sensation. Next function is control of the autonomic nervous system. Control of the autonomic nervous system. Nerve fibers arise from cerebral cortex, amygdala, parts of limbic system, reticular formation, and these nerve fibers accompany reticulospinal tract to go to the spinal cord, and where these nerve fibers terminate onto the preganglionic sympathetic neurons, terminate onto the neurons which are preganglionic sympathetic in segment T1 to L2, and these nerve fibers also go to the neurons which are preganglionic parasympathetic. So these are terminate onto neurons which are preganglionic parasympathetics in the segments S2, 3, 4. In the segment S2, 3, 4. Next function is that reticular formation is involved in the control of anterior pituitary hormone secretion. Involved in the control of anterior pituitary hormone secretion. Reticular formation has connections with nuclei, which secrete releasing and inhibitory hormones from the hypothalamus. So reticular formation has got connections with nuclei in the hypothalamus which secrete releasing and inhibitory hormones. So through these releasing and inhibitory hormones, reticular formation controls the secretion of anterior pituitary hormone secretion. Next function is that reticular formation has a role in the control of circadian rhythm or biological clocks. It has got a role in the control of circadian rhythm or biological clocks, because reticular formation has connection with the hypothalamic nuclei, especially suprachiasmatic nucleus, which control the biological clocks or circadian rhythm, such as diurnal variation in the body temperature, secretion of ACTH, corticosteroids, secretion of melatonin, cycle of the sleep and wakefulness. So these are the biological clocks or circadian rhythm. These are all controlled by the hypothalamus basically, and reticular formation by connection to hypothalamic nuclei controls the biological clock or circadian rhythm. Next function is that reticular formation has the reticular activating system. As the reticular activating system, which maintains wakefulness or alertness, which maintain wakefulness or alertness. So this was about the functions of the reticular formation. Now briefly about the connections of the reticular formation. Now first about the afferents which go to reticular formation. So afferents for spinal cord as spino-reticular tract, then collaterals from the medial meniscus, spinothalamic tract, medial meniscus, spinothalamic tract, visual, olfactory, gustatory, vestibular pathway, collaterals from visual, olfactory, auditory, gustatory, and vestibular pathway. Then afferents from the cerebellum, thalamus, hypothalamus, subthalamus, amygdala, from the motor areas, from the sensory areas, right? So these are the afferents. Now efferents from the reticular formation. Efferents from the reticular formation, these go to spinal cord as reticulospinal tract. Other efferent are the reticulobulbar tract, then efferents to thalamus, efferent to corpus striatum, to subthalamus, hypothalamus, amygdala, parts of the limbic system, substantia nigra, then efferent to the motor cortex, to the sensory cortex, efferent to the motor and sensory cortex. So this was about the connections of the reticular formation. Keep in mind that alcohol, barbiturates, and general anesthetics depress the activity of the reticular activating system. Alcohol, barbiturates, and general anesthetics, these depress the activity of the reticular activating system. Adrenergic drugs or sympathomimetic drugs, adrenergic or sympathomimetic drugs, these excite the reticular activating system, resulting into alertness, insomnia, loss of sleep, or alertness or wakefulness. So this was about the reticular formation. In the brain, there are certain neurohormonal systems. In the brain, there are certain neurohormonal systems. So I discussed neurohormonal system in the brain. So there are 4 neurohormonal systems in the brain. Now, these neurohormonal systems in the brain, the first one is Raphael nuclei and serotonin system. Raphael nuclei and serotonin system. Raphael nuclei are midline nuclei at the junction of the pons and medulla oblongata. So midline nuclei at junction of the pons and medulla oblongata. Nerve fibers from Raphael nuclei, these go to the reticular formation, hypothalamus, thalamus, and many other parts of the brain. Nerve fibers from the Raphael nuclei also go to the pain inhibitory complex in the dorsal horn of the spinal cord. So nerve fibers from Raphael nuclei also go to the pain inhibitory complex in the dorsal horn of the spinal cord. Dorsal horn of the spinal cord. You know, serotonin is involved, along with the Raphael nuclei, is involved in the deep sleep, and also Raphael nuclei are part of Energizing System. Also, Raphael nuclei are parts of Energizing System. Serotonin is the neurotransmitter released by the nerve fibers arising from the Raphael nuclei. So serotonin is the neurotransmitter released from the nerve fibers arising from the Raphael nuclei. So this was about the first neurohormonal system, that is Raphael nuclei and serotonin system. The second system is locus ceruleus and norepinephrine system. Locus ceruleus and norepinephrine system. Locus ceruleus is located in the lower midbrain and upper pons. Located in the lower midbrain and upper pons. Nerve fibers from this locus go to reticular formation and other parts of the brain. Nerve fibers from locus ceruleus, these go to the reticular formation and other parts of the brain. Norepinephrine is released at the nerve endings of these nerve fibers. So these nerve fibers arising from locus ceruleus release norepinephrine at the nerve endings. And you know, norepinephrine is involved in alertness or excitation. This locus is involved in the REM sleep, rapid eye movement sleep. Involved in REM sleep or rapid eye movement sleep. This was about the locus ceruleus and norepinephrine neurohormonal system. The third system is gigantocellular neurons and acetylcholine system. Gigantocellular neurons and acetylcholine system. These neurons are very large neurons located in the reticular formation of the upper brainstem. So very large neurons located in the reticular formation of the upper brain stem. And these neurons release acetylcholine. These neurons release acetylcholine at the nerve endings, at nerve fibers. These release acetylcholine. So these neurons are also involved in the excitation of the brain and also in REM sleep, rapid eye movement sleep. So these neurons along with acetylcholine are involved in the brain excitation and also in the REM sleep, rapid eye movement sleep. The fourth neurohormonal system, is substantia nigra and dopamine system. Substantia nigra and dopamine system. You know, dopamine is an inhibitory neurotransmitter. Dopamine is an inhibitory neurotransmitter. Nerve fibers from substantia nigra. Nerve fibers from nigra which go to the caudate and putamen, release dopamine at the nerve ending. We say that nigrostriate fibers release dopamine at the nerve endings. Now dopamine along with acetylcholine is involved in the control of the body motor activity. So dopamine along with the acetylcholine is involved in the control of the motor activity. Now when there is degeneration in substantia nigra, so neurons in nigra when degenerate, it results into Parkinson's disease. So degeneration of neurons in substantia nigra, it results into Parkinson's disease. In this disease, there is dopamine deficiency in caudate and putamen. In this disease, there is dopamine deficiency in the caudate and putamen. This results into features of the Parkinson's disease. So this was about the substantia nigra and dopamine neurohormonal system. So I have discussed 4 neurohormonal systems which are present in the brain. Thank you.